Hello, my name is Panagiotis Lalagkas. I am a graduate student at the University of Massachusetts Lowell, and today I will talk about drug repurposing and how we can leverage Mendelian and complex disease comorbidity for this purpose. Why is drug repurposing important though? To answer this question, let's consider the traditional drug design pipeline. It involves preclinical studies, where researchers identify a target molecule or a pathway for a specific disease. They design a drug that targets it and conduct in vitro and in vivo experiments. Then, the candidate drug goes through clinical trials to assess safety and efficacy. And finally, if everything goes well and the usefulness of this drug for the disease under study is proven, the drug receives approval and is released in the market. However, this process takes approximately 12 to 15 years, and millions or billions of dollars are spent in order a single drug to be approved. So we need a way to reduce the time and the cost. And this way might be drug repurposing. So drug repurposing refers to the process of finding new uses for an already approved drug. That means that the drug under study is known, so preclinical studies are not required. Also, because we talk about approved drug, we know that it is safe for use in humans, so phase 1 may be skipped too. And because of these, drug repurposing takes approximately 3 to 6 years only. That means that the cost is reduced, and also the drug reaches the market faster, which eventually benefits the patients. Among others, genetics has played a significant role in drug repurposing. Let's see it through an example. Let's say we have one group of people with Crohn's disease and a second group of people without Crohn's disease. We can genotype them and perform a genome-wide association study, a GWAS, to identify mutations or SNPs that are found more frequently in the group of people with Crohn's disease. If we do this, we will find a signal in the locus of the IL-23R gene, meaning that mutations in this gene are associated with Crohn's disease. Having this knowledge, we can look for drugs targeting this gene. Ustekinumab, a monoclonal antibody originally approved for psoriasis, is one of them, as it targets the P40 subunit of the IL-23. So based on that, we can hypothesize that this drug might be useful for the treatment of Crohn's disease. And indeed, ustekinumab has been tested in clinical trials and nowadays is also approved for the treatment of Crohn's disease. So this is an example of how genetics can inform drug repurposing. However, in order this to be a successful approach, it requires the correct mapping of the identified SNPs from the GWAS to the causal genes. And this is not always easy. This is mainly because the majority of GWAS hits are in non-coding regions, which makes the mapping of identifying SNPs to the causal gene a difficult task. So we need another way to learn about disease biology. Mendelian disease comorbidity can be an alternative way. I will use the example of the Rubinstein-Taybi syndrome to show you how. So in general, Mendelian disease causal genes are very well known due to their high penetrance. So in this example, we know that Rubinstein-Taybi syndrome is commonly caused by mutations to the CRBP gene. Also, from studies that analyzed electronic health records, we know that a lot of these patients get lymphomas. Lymphomas are not usually inherited like Mendelian diseases, but are caused by acquired mutations. And one of the currently inactivated causal genes is the CRBP as well. So here we can see that maybe the inherited inactivation of the CRBP gene in people with the Rubinstein-Taybi syndrome is what causes this predisposition to lymphoma. So this example shows how Mendelian disease comorbidity can tell us about the underlying biology of a comorbid complex disease. In the work I present today, we go a step further and we ask if Mendelian disease comorbidity can also be used for drug repurposing. Let me introduce you to the hypothesis we make. So from the previous slide, we saw that Rubinstein-Taybi syndrome is clinically associated with lymphoma. Also, we know the gene that causes the Mendelian disease. And moreover, from previous slide, we saw that the Mendelian disease comorbidity can be informative about the underlying disease biology of the complex disease. That means that the Mendelian disease causal gene can be suggested as drug target for the lymphoma. And based on that, we find drugs targeting that gene and we recommend them for repurposing for lymphoma. In other words, we hypothesize that drugs targeting the Mendelian disease causal genes can be repurposed for the comorbid complex disease. So in general, let's say we have a Mendelian disease 1 and 2 and their corresponding comorbid complex diseases 1 and 2. We know the Mendelian disease causal genes and we find drugs targeting them. Based on our hypothesis, the blue pill might be useful for the treatment of complex disease 1 and the orange pill for the complex disease 2. To test it, we collect information about drugs already investigated or indicated for each complex disease. So in order our hypothesis to be correct, we want the drugs we recommend for repurposing for a complex disease to be also in the list of already investigated or indicated drugs for that complex disease. So we recommend the blue pill for complex disease 1, for example, and we want it to be also in the list of the investigated or indicated drugs for that complex disease. To test our hypothesis, we use a logistic regression approach where we ask if the drugs we recommend for repurposing for a complex disease are more likely to be investigated or indicated for that complex disease. And we also account for the number of drug targets and the disease category in our model. So, the significance is assessed by the coefficient of the independent variable recommended in our model. To test the strength of significance, we do permutation tests. That means that we randomly shuffle the comorbidities of the Mendelian diseases. So, for example, after a random shuffle, let's say that Mendelian disease 1 is comorbid with complex disease 2 and Mendelian disease 2 is comorbid with complex disease 1. Based on our hypothesis, we would recommend the blue pill for repurposing for the complex disease 2 and the orange pill for the complex disease 1. We then run the logistic regression I mentioned before and we obtain a p-value. We do this 1000 times to create a distribution of p-values to which we compare the original p-value before the permutations. So here you can see in blue color the distribution of the p-values we obtained after 1000 permutations. The x-axis is the negative log 10 of the p-value, so the significance increases from left to right. The red dashed line corresponds to the p-value of 0.05 and the black dashed line to the observed original p-value. As you can see, the observed p-value is more significant than the p-value of 0.05, meaning that the drugs we recommend for repurposing based on Mendelian and complex disease comorbidity are more likely to be investigated or indicated for the comorbid complex diseases. Also, the observed p-value fall after the right tail of the distribution of the permuted p-values, suggesting that the observed p-value is unlikely to occur by chance. So yes, Mendelian disease comorbidity can be used for drug repurposing. We also performed the same analysis as before, but we evaluated our recommendations using information about drugs being investigated for the complex diseases and are in either phase 1, 2, or 3, or drugs that are already indicated. Again, the colored areas are the distributions of the p-values obtained after 1000 permutations by shuffling the Mendelian disease comorbidities. The x-axis is the negative log 10 of the p-value, and the black dashed line is the observed p-value in each case. As you can see, the drugs we recommend for repurposing are more likely to be investigated in phase 2 or 3 or to be already indicated for the comorbid complex diseases. Also, we can see that as we go from phase 2 to phase 3 and to indicated, the significance increases as the black dashed lines move to the right, meaning that our recommendations are more likely to be in advanced stages for the corresponding complex diseases. And as you can see from the distribution of permutations, this is not something we would expect by chance. However, not all the Mendelian disease-associated genes are targeted by the same number of drugs. Some Mendelian disease genes might be targeted by one drug and other by 10 drugs. So this led us to investigate if there is any relationship between the number of drugs targeting each Mendelian disease genes and the usefulness of that Mendelian disease for drug repurposing. So first, we grouped the Mendelian diseases into non-significant and significant by running the same logistic regression as before, but for each Mendelian disease separately this time. After we formed the two groups, we found that Mendelian diseases in the significant group, which is shown in blue color in the figure, are targeted by statistically significantly higher number of drugs compared to non-significant Mendelian diseases. So this is a very interesting finding. Although we don't know yet why this is happening, we suspect that the genes associated with the Mendelian diseases in the significant group might be more well studied, and that's why there are more existing drugs targeting them. Or it might be that the genes associated with the Mendelian diseases in the significant group are better drug targets than the others, and that's why more drugs are available for them. Moreover, for some of the Mendelian disease and cancer comorbidity pairs we examined, we had information about genetic similarity from a previous study. Let me explain to you what I mean by saying genetic similarity. So briefly, Melamed et al. Took the 10 cancers that show comorbidity with 60 Mendelian diseases, and for each one of them, they created a gene set containing the most commonly mutated genes. Also, they had the genes associated with the 60 Mendelian diseases. Then, they established 4 metrics to assess the genetic similarity of the comorbid pairs. For example, one of the metrics was the gene enrichment, where they tested if the comorbid diseases share the same causal genes. They also looked for shared pathways, direct interactions using a network approach, and co-expression of the Mendelian disease genes and cancer genes. So, in this project, we got this information and we asked if comorbidity pairs with genetic similarity are more useful for drug repurposing. So, we performed the same analysis as before, but we only used the Mendelian and cancer comorbidity pairs with or without genetic similarity. You can see the observed p-value indicated by the blue dashed line and the distribution of the permuted p-values in blue color. As you can see, the results are significant, meaning that Mendelian disease and cancer comorbidity can be used for drug repurposing for the cancers. Next, we used only the Mendelian disease and cancer comorbidity pairs with genetic similarity, indicated by the brown color. And as you can see, the significance increases, suggesting that genetic similarity of comorbid diseases might enhance drug repurposing opportunities. So in summary, I hope I convinced you that Mendelian disease comorbidity can be used for drug repurposing for the comorbid complex diseases, and that drug repurposing might be enhanced by focusing on Mendelian diseases targeting by high number of drugs, and by focusing on comorbidity pairs with genetic similarity. At this moment, I would like to thank my supervisor, Dr. Rachel Melamed, for her guidance and motivation, and the members of the Melamed Lab for the insightful discussions we had. Also, I would like to thank the funding sources, the National Institutes of Health, and the Department of Biological Science of UMass Lowell. And of course, I would like to thank you for listening.